So welcome to La Jolla Institute. Thanks for coming to hear me talk tonight about the immune systems, Dr. Jekyll and Mr. Hyde. So in terms of tonight's topic, we'll start off simply. What is cancer? So normally the cells in our body know exactly when to stop growing. So they grow, they divide, and they stop. And they know when to stop. But in all types of cancer, our body's normal cells forget how to stop growing. They continue to grow, and then they can actually, in the case of a malignancy, spread into surrounding tissues. Now a little introduction to immune cells. So I think many of you have come to these LGI Life Without Disease events before, so you've heard a lot about immune cells, particularly T cells. Overall, these cells function to keep us healthy. So if we didn't have an immune system, we wouldn't be sitting here. They really are important in protecting us from pathogens like bacteria, viruses, and even cancers. They help us heal when we get an injury. So if we cut our hand or we break a bone, they help us heal. But they can also be tricked by cancer cells to actually help the cancer grow. And I'm going to tell you about that tonight. If we can figure out how to stop cancer cells from evading the immune system, we can cure cancer. So what are immune cells in cancer? Well, there are two types or two classes of immune cells. Myeloid cells, so now this is where you're going to remember what my title meant, and lymphoid cells. So myeloid cells are these cells: macrophages, dendritic cells, monocytes, which is one of my favorite cells, and neutrophils, which is another favorite cell. So when you look at myeloid cells, think of Pac-Man. Do any of you remember that game where that yellow dot, would go and get the dots and the ghosts? So this is kind of like a myeloid cell. They kill first and ask questions later. If something looks bad, they kill it, right? And then their other job is to alert lymphocytes of danger. So what's a lymphocyte? A lymphocyte is part of this lymphoid cell class, the other class of immune cells. These are B cells and T cells, and I know if you've come to these events before, you've heard a lot about both of these from my wonderful colleagues. These cells act more slowly to kill specifically. So these guys kill quickly and not very specifically. They just see something bad and they go for it. These guys kill very specifically. They take a lot longer to plan an attack. They're given signals by these cells. So these cells tell these cells kind of what to do. The lymphocytes are much more strategic in their war game, but we need them both. And the important key is that they both talk to each other. So now we get into Dr. Jekyll and Mr. Hyde. So we'll specifically now for the rest of my talk really going to talk more about the myeloid cell branch, this innate immune branch of the immune system. These cells are important healthy parts of our immune system, but they can actually play different roles in cancer. Myeloid cells are this way. Some are Dr. Jekyll, they're good guys. Some are Mr. Hyde, they're kind of good guys, but they turn into bad guys. So first we're going to talk about non-classical monocytes. And a lot of what we do is we try to work in live mice to actually image how these cells function and how they interact with cancer cells. And to do this, we study mice that express a green fluorescence selectively in non-classical monocytes. So in other words, if you put this live mouse under a microscope, then you can actually see these green non-classical monocytes, and we can actually study them. And I'll show you some movies. A few years ago, we made this really, really important discovery in the lab. And that is that these non-classical monocytes, these green cells, actually can search, find, detect, and help kill cancer cells. So this is an image from a live mouse from a lung. The red here is the tumor, and the green are the non-classical monocytes. And what you can see here is that the green cells, the non-classical monocytes, are circling around the tumor cell. So, and you can, if you watch this guy, you can see he actually goes and he's touching the tumor and trying to sense it, maybe trying to eat it. And you can see this here. This one is going to go chase this tumor cell. We found that they actually recognize and help kill the cancer cells. And in doing so, they prevent metastasis. So this is your first line of defense. So again, this is a still microscopy image Again, the non-classical monocytes are shown in green. The tumor is shown in red here. And what we found was that in yellow here, the non-classical monocytes, the green cells, actually take up the tumor pieces. So they're actually helping, they seize the tumor cells, they help kill the tumor cells, they take up the tumor debris, and then they actually, remember the lymphoid cells, they actually now take these little pieces of tumor to an organ called the lymph node, and they actually then help give this tumor cell information to the T cell to activate that T cell. So remember I said there's two arms, and that T cell takes a little bit longer to respond because this cell and some of its cousins are going to the lymph node. They're telling the T cell, look, here's a tumor. You got to go out here and help get rid of this thing. Now I'm going to tell you a story about the CD33 positive monocyte. And to do that, I have to mention immunotherapy. So I think most everyone in here has heard of immunotherapies. These are revolutionary cancer treatment breakthroughs. Two of them are pretty important and very new. It's something called CAR-T, which stands for chimeric antigen receptor T cell. And I'm going to ask you, Nan, what that is later. So just think CAR-T. And this is where a person's own T cells are engineered to recognize cancer cells. The other therapy is checkpoint. It's called checkpoint blockade. And these are antibody therapies that actually prevent cancer cells from turning T cells off. So these are revolutionary therapies that are really changing the way cancer is treated in the world. So what are the responses to these immunotherapies? No matter how successful they've been, and they've been extremely successful, is that the response rates to these therapies really are only about 30% of patients. So we're interested, and many of us in the cancer field are really trying to understand why this is, right? So I'll touch on this one. Can we identify which patients may respond before they get treatment? And this is where the CD33 monocyte comes in. And this is work from an outstanding postdoc in the lab, Claire Alinghi, who's sitting in the back. And it's in collaboration with Sandeep Patel at the Moores Cancer Center at UCSD. And so what Claire did was we obtained from Sandeep, Dr. Patel, patients that have been given this PD-1 checkpoint blockade. So they have been given this antibody therapy that I described. And what Dr. Patel knew was that some people did not respond to the therapy. So their cancer actually progressed. But then he had some patients that did respond, so their cancer stopped progressing. And so Claire took blood from the patients, and she did this method called mass cytometry, which is immunoprofiling of immune cells in these patients. And what this is she was able to take this big panel of markers, and these are all markers of proteins that sit on the surface of cells. So by doing this huge panel, we can identify what immune cell populations are in these patients' blood. And Claire could ask what cell types are different in people that respond versus people that don't respond. So that's what Claire has set out to do. You can imagine with all those markers that this is a lot of data, right? And it's hard for us to really analyze it. So we use computers, and what we get are little plots that look like this, that you're thinking, well, this tells you nothing, right? Well, let me tell you, each dot here is a cell from a patient's blood. The computer analysis kind of groups clusters or cells that are similar together. So in other words, this group of cells have some of the same markers floating on the surface, so they're grouped together, whereas this group down here has different markers on its surface, so they're grouped together. So by understanding what markers these all are, we can ask the question, are there cell types that are present in a responder that are not present in a non-responder, or vice versa? So this gives us an idea of what cell types may play a role in regulating whether someone responds to checkpoint therapy. So Claire did this, and what you can see is on the left is a non-responder and on the right is a responder. So what you can immediately see in blue here is that there are populations of cells in the responder's blood that are not present in the non-responder. So you want to know, well, what are these? Because maybe these are important for why this person's responding to therapy and why this person is not. So Claire figured this out. She knew the markers, and guess what? One of the markers is our green non-classical monocyte that we showed you before prevented metastasis. We had no idea that it would show up in a responder, so this is new data. And she's graphed the cells, and here the responders have more of them than the non-responders. So now I'll just tell you briefly about myeloid cells that can become Mr. Hyde. So these are good guys that go bad. And these cells are classical monocytes, which is a cousin of the non-classical monocyte, macrophages, and neutrophils. And these three cells can actually be tricked by cancer cells. The normal functions of these cells are either exploited by the cancer cell, or the functions of these cells are changed completely by the cancer cell. So these cells are really, really important for maintaining health and having a healthy body. Neutrophils are known to be important very early, like if you cut your hand, they're the first little cell in there that really helps clean the wound before it gets bacteria in it. So these are very good cells, but they definitely become Hyde because they help cancer cells grow. And they also are the cells that actually can inhibit immunotherapies, whether it's a checkpoint blockade or whether it's a CAR T, chimeric antigen receptor T cell. And so how are these cells tricked, right? So the cancer cells trick the myeloid cells to secrete proteins that turn off the T cell or turn off the CAR T cell so that it doesn't work. And these cells also provide nutrients to actually help cancer cells grow. They can actually secrete nutrients that actually help the tumor grow blood vessels, so it helps get nourishment and can grow bigger. They also can actually secrete things that actually help degrade surrounding tissue so that the cancer cell can metastasize and migrate through the tissue and grow and expand. And so in this way, these cells actually are quite Hyde-like. Thank you.